Cancer has been the leading cause of death worldwide, accounting for 8.2 million deaths around the world in 2012. Everyone surely knows that cancer is deadly, but not all of the reasons are made known yet, and the genetic reasons will be explained here. In fact, 5% of all genetic mutations are hereditary, while the other 95% originates from life habits and environmental causes. Also, it takes 6 mutations per gene for the gene to be random mutated. Let's discuss the different genes found in the DNA in each cell. They control how the cell functions, how quickly it grows, and how often it divided, and how long it lives. Our genes pick up mistakes that occur when cells divide. These mistakes are called faults or mutations that happen throughout our lives. They are caused by natural processes in our cells and by various other factors. These include tobacco smoke, radiation, ultraviolet radiation from the sun, some substances in food and chemicals in our environment. Sometimes people inherit faulty genes from their parents. Usually, cells can repair faults in their genes. If the damage is very bad, they may induce apoptosis instead, or the immune system may recognize them as being abnormal and kill them. This helps to protect us from cancer. But sometimes mutations in the important genes can mean that a cell no longer understands its instructions and starts to multiply out of control. It doesn't repair itself properly and it doesn't die when it should. This damages neighboring cells and ultimately leads to cancer. There are 4 main types of genes involved in cell division. The first type is oncogenes. They encourage cells to divide and multiply. The second type is the tumor suppressor genes. They do the opposite. They stop the cell from multiplying. The third type produces the repair proteins for the cell's damaged DNA. The fourth type are the genes that induce apoptosis. Most tumors have 40 copies of more than one of these types. Oncogenes are genes that, under normal circumstances, play a role in telling cells to start multiplying and dividing. HER2, a protein synthesized from oncogenes, controls cell growth and spread. The RAS family of genes make proteins involved in cell communication pathways, enabling apoptosis and cell growth to go smoothly. Usually, cells can repair faults in their damaged genes, but if the damage is very bad, these genes may stop the cell from growing and dividing. The best known tumor suppressor gene is p53. Mutations in the p53 gene, amongst all other tumor suppressor genes as well as oncogenes, lead to the cell no longer understanding the message from the gene to stop growing and multiply out of control. Then, over generations, cells will become too small to function properly. This rapid division is one of the reasons why cancer spreads so quickly to other organs, leading to more cells with damaged DNA in the body, making it so deadly. The DNA in every cell in our body is constantly in danger of being damaged. However, cells contain many different proteins whose job is to repair damaged DNA. BRCA1 and BRCA2 are human genes that produce the DNA repair proteins. These proteins help repair damaged DNA and therefore play a role in ensuring the stability of the cell's genetic material. But if the DNA mutations occurs to BRCA1 or BRCA2 amidst all other types of DNA repair genes, this protein product either is not made or does not function correctly. DNA damage may not be repaired properly. As a result, cells are more likely to develop additional genetic alterations that can lead to cancer. The last type, the genes enabling self-destruct. In the case of apoptosis, these genes normally instruct a cell to self-destruct if it has become too old or damaged. However, once these genes are mutated, the instructions for the cell to die become unclear. As a result, the cell could no longer follow the instructions of the defective resultant self-destruct protein and continues to live on. As mentioned earlier, this leads to many more defective cells, which accounts for cancer ultimately. Today, we have learned the different types of genes that play a primary role in the cell's life cycle. Mutations to these genes, either hereditary or from external factors, can lead to the genes and the resultant proteins to be unable to carry out their function adequately. As it takes 6 mutations per gene for it to be considered mutated, considering that there are 4 types of genes regulating cell division and death, there will be at least 24 mutations if all 4 types of genes are mutated. When accumulated, this renders the already damaged or dysfunctional cell to continue living, and this can lead to cancer.